from the ancestor at high CO2. Nothing happened, right? But when we look at the reciprocal transfers, we see some interesting things. First off, if you take the low CO2 adapted ones, switch them over to high CO2, they almost immediately go back up. And again, that's just a short-term experiment like the ones I showed you. But look at what happens to the 750 high CO2 adapted ones when you take them back down to low CO2. They still are fixing nitrogen as if they're in a future CO2 atmosphere, and now they're back down at low CO2. We scratched our heads and said, what's this all about? How can they do that? So we said, let's keep them going for a while and see how long it takes the rate to decay back down to present day levels that we see, say, down here. So we did that. We took this strain, and then we've kept going with our 380 adapted and our 750 adapted. And I'm going to show you that in a minute here. So this is just summarizing what I just said. The N2 fixation rates got stuck at those high rates even when we moved them back. So this is the same cultures for over a year after that reciprocal transfer. This is simply the 380 adapted cultures. And this is the high CO2 adapted cultures. As I told you, they're just kind of growing along. Let me show you the one that was switched now from high CO2 back down to this concentration. It's still fixing nitrogen just like it's in the high CO2 world. Even though it's growing now for almost 500 days, actually it's quite a bit longer now. This graph doesn't take in all of our data. It's now been growing for almost 2 years at low CO2, and it's still fixing nitrogen as if it's at high CO2. Just to convince you that this is a real thing, this constitutive upregulation of nitrogen fixation, and that happened in all of the cell lines, 6 replicates, every single one did it. But just to convince you, these are the growth rates. Again, the low CO2 adapted in yellow, the 750 ppm adapted, and in blue, the 750 switched back to 380. So they're growing as if they're at high CO2, but they're now back at low CO2. But microbial evolutionary biologist who looks at this and says, growth rates, that is the currency for fitness in a microbe, for reproductive success, right? This means that there's been a 32% increase in the fitness of those cultures from being selected at high CO2. But the really odd thing is this 32% increase in fitness didn't happen in the novel environment we're selecting them in. It happened in the ancestral environment. As far as we can tell, this result is unprecedented in the evolutionary literature. Never does an organism become more fit in the ancestral environment when you evolve it in a novel environment. It's never been seen before. So what's going on? We decided that we really had to try to figure out why they were doing this. So in terms of summarizing the phenotypic responses of trichodesmium after long-term selection by high CO2, at high CO2 you don't see any response. They don't get more fit, they don't grow faster at high CO2. What you see is they're now more fit when you move them back to low CO2 because they're still growing and fixing nitrogen as if they were in the CO2 world. And so we can define what we call a high CO2 phenotype. So that consists of high nitrogen fixation and growth rates. And in our experiment, if you think about it, there's 3 treatments that have this phenotype. There's the 750 adapted ones. There's the 380 ones that will switch over to that if you move them into the high CO2 very quickly. And then there's these weird guys, these 750 adapted cell lines switch back to 380. They're all exhibiting this high CO2 phenotype. So how can we use our omics data, or in this case the transcriptome data, to try to get a handle on what's happening here? Well, we reasoned that we should focus on finding genes that are expressed in the same treatment-related patterns as the phenotype. So we want to see genes that are upregulated in the 750 adapted and the 380s when you move them to 750, but then they aren't downregulated again when we move the 750s back. In other words, they're shared by all of the high CO2 phenotype cell lines, just like the nitrogen fixation and growth results. So I'm not going to show you the genes, all of the genes that did this. I'll show you a little summary slide because there's a lot of them. What this is it's a metabolic map, which you can't see very well, of trichodesmium, but it doesn't matter. And this shows you all the up and down regulated pathways in the transcriptome in these various treatments. So the green ones are the ones that were upregulated in 750. And what you can see, there's some amino acid metabolism pathways, oxidative phosphorylation, glycolysis, pentose phosphate pathway. Interesting. So what does that mean? There's also some that are downregulated in the 750 adapted cells. You can't see this very well, but here's purine metabolism and iron metabolism. But the ones we're particularly interested in are the ones that all three of those treatments share. And those are these two. Genes involved in photosynthesis and carbon fixation. Not nitrogen fixation, photosynthesis and carbon fixation. As a matter of fact, we didn't see any response of things like the nitrogenase genes that we expected to see stuck on in the high CO2 switch cell lines. So the pathways that are upregulated in the cell lines with this high CO2 phenotype, well, what's conspicuously lacking is any response of nitrogenase. There doesn't look like nitrogen pathways are what's changing. What actually changed were the cell's energetic pathways, things like photosynthesis, carbon fixation, oxidative phosphorylation. So what's happening is they're bringing in more energy to drive nitrogen fixation. They're not actually synthesizing more enzyme. I'm going to switch gears now because we saw something else interesting in the transcriptome that looked like a side issue, but we think maybe it's not. And to tell you about that, I have to give you a little background. Trichodesmium, which is a perfectly good photosynthetic bacterium, has a really weird bacterial genome. Here's trichodesmium up here. These are genes. Here's 4 other cyanobacteria. And if you look closely, what you can recognize, you don't have to look too closely because I've circled them, is there's empty spaces in the genome of trichodesmium that don't code for any proteins. In other words, for a prokaryote, the genome of trichodesmium is littered with intergenic DNA that doesn't code for anything. It's almost like a eukaryote in that respect. 40% of the genome is non-coding. So what's going on here? This is a paper that our student just published a couple of weeks ago. Well, one way to look at it is this. Let's look at the sequences that were upregulated in the 380 to 750 switch, but then not downregulated. We'll use that same method in the 750 to 380 switch. The same pattern as nitrogen fixation. So ribosomal transfer RNAs, 8 were upregulated with that pattern. Not a surprise, they're growing faster. They need to carry around amino acids for protein synthesis. There were 77 different genes that showed that pattern. Look at the intergenic regions. 140 intergenic regions were upregulated in those stuck cell lines. What's going on? Well, we looked at what these intergenic regions are, and Nate did a very nice job. This is his work. Some of those are known regulatory regions, such as promoters, that don't show up in proteins. That's not unexpected. But a lot of them are not. They are expressed, however, as small non-coding RNAs, which we confirm by northern blot analyses. So they make little RNAs that fold up. And interestingly enough, these NC non-coding RNAs have been shown to regulate gene expression during stress responses in a lot of other bacteria. So one of the responses to being selected at high CO2 is to upregulate all these non-coding regions. In other words, those expressed intergenic regions are not junk DNA, they actually seem to be performing a function. And we think that role is controlling the responses of trichodesmium to environmental change. We've also seen some other interesting responses in these weird stuck in the on position, stuck in the fast lane cell lines. This is a photo period from when the light goes on to when it goes off, nitrogen fixation rates. And this is normal for trichodesmium. It fixes nitrogen during the light. Here's the 380 selected, the 380 selected cell lines here in red. And this is pretty normal. Nitrogen fixation peaks between, say, 3 and 5 hours in the photo period and then declines toward the dark period. Here's the 750 ppm adapted, the yellow ones. And what you can see is they have a much longer peak period, more like 5 or 6 hours than a couple, and it's shifted to later in the afternoon. And our 350 to 380 ppm switch cultures that are stuck in the on position, their diel pattern has also become stuck in this position. So this is a couple years after they've been moved back to low CO2. They're still showing a diel pattern that resembles that of the high CO2 adapted cell lines. So one of the things we're looking at right now is clock genes and seeing if they have something to do with this. Cyanobacteria are the most primitive organisms that have a true circadian rhythm. And there's a lot known about their clock genes. And this is one thing we are looking at. And then finally, another thing we noticed in these cell lines that really sticks out is we did some enzyme activity assays. This was based on something we saw in the transcriptome. I'm not going to show you that. What we did is we bought a kit that examines methylation of cytosine residues in DNA. And in eukaryotes, it plays a role in epigenetic gene expression. Is very common. So the DNA is methylated, genes can be turned off that way, and perhaps on as well. Look at how high the activity is in this weird set of 750 to 380 switch cell lines compared to the other two. So in other words, there's a lot of methylation of DNA going on. And we're pursuing this to try to figure it out. Right now, we're running bisulfite sequencing on the genomes, which will show us which parts of the DNA are methylated. And we're also very interested because prokaryotes aren't supposed to do this. It's another unbacterial-like response that we've seen. So in conclusion, for this whole section, is both the evolutionary mechanisms that I mentioned in the beginning seem to be important potentially for adaptation of these N2 fixers in the future high CO2 ocean. There's a lot of standing genetic variation that already exists that will allow CO2 to select for particular strains. And elevated CO2 can also select for new and, in many cases, very unprecedented, at least in the literature, responses of physiology and genetic and perhaps epigenetic responses in these individual cell lines. So de novo evolution is going on, but it's certainly not what we expected. We thought they would get better at growing at 750. Instead, they got better at growing at low CO2. So where does this leave us with this biogeochemically critical organism? What are the implications? Well, you can probably think of some, but one question I have is if trichodesmium is going to irreversibly adapt to high CO2 levels, what happens if we ever get our carbon habit under control and actually lower the CO2 in the atmosphere? Can it readapt back? Well, I would assume long enough, maybe it will, but our cultures have not done it in several years. They're stuck. Another question is, can this help explain why we see so much variability in these strains? Did these strains get stuck at some state in some past high or low CO2 level of the atmosphere? Is that why we see strains that are adapted for these two types of conditions? And then finally, I think one lesson that's an obvious and an easy one is we've been studying trichodesmium for 30 years. It's without a doubt the most studied marine nitrogen fixer. And yet, look at this evolutionary surprise that it's given us. We don't understand much at all about what it's doing, about how it regulates its genes, about how it responds to evolutionary changes. And we say it's well understood, but we don't understand it. And so you can still get a PhD out of a well-studied organism. For the rest of my talk, I want to talk about this. What about iron and phosphorus limitation? It's an obvious question. So won't the iron and phosphorus limitation just kind of trump this increase in nitrogen fixation we see by high CO2? And to some extent, it does. I'll show you. But there's, again, more to the story. And before I show you the CO2 part of it, I want to show you some work we've been doing with iron and phosphorus limitation of trichodesmium. Just at normal CO2, we have not selected these. These are not my selected cell lines. What we're finding is that they would rather be limited by both iron and phosphorus than limited by either one alone. They are better adapted to be co-limited by iron and phosphorus than to be unlimited by just iron or just phosphorus. Here's the nitrogen fixation rates of replete cultures, plenty of iron and phosphorus. Iron limit them, those get low, right? Oops. Somewhere here, I keep doing that. Here's the phosphorus limited ones. Of course, they're much lower. But look what happens when you limit them for iron and phosphorus. They can't recover all the way up to the replete ones, but they can grow much faster than cells that are grown either with iron or phosphorus alone limiting them. So co-limitation appears to be an advantage. If you're going to be limited, you might as well be limited for two things. Let me talk to you about that for a minute. This is a paper one of my graduate students published last year. Nathan Garcia, who's now with Adam Martini doing a postdoc there. So he grew both Crocosphaera and trichodesmium across a range of phosphate concentrations. And the dark symbols are iron replete, the light symbols are iron limited. And what you can see is that high phosphorus concentrations, as you expect, the ones with iron, greatly outgrow the iron limited ones, just like you expect. But look what happens when you start getting down to very low and limiting phosphorus concentrations. The lines cross. The iron limited ones crash about here. The iron replete, the iron limited ones can keep growing to much lower phosphorus concentrations way down here. And we see that in both of these fairly distantly related genera. How are they doing that? Well, we had a theory because we noticed something. The ones that are co-limited by iron and phosphorus down here are smaller. They miniaturize. And I'll show you that. How do they do it? They get smaller. This is Crocosphaera. This is the cell volume, again, in the iron replete ones and in the iron limited ones across a range of phosphorus. And you can see when they have a lot of iron, the cells stay the same size, but when they don't, when they're iron limited, they get smaller and smaller. And what's going on here? Well, what we think is when they're limited by both, they're able to miniaturize and they can keep their specific growth rates higher because they need less carbon, less nitrogen, and less phosphorus to build a new cell. And so they can grow faster. Their specific growth rates are faster. I'll come back to this again in a little while. So let's get back to the CO2. How's that interact with this? This is the growth and nitrogen fixation rates of those same cell lines I was talking about earlier, the high and low CO2 adapted trichodesmium, which we then grew under iron limitation, phosphorus limitation, or co-limitation. So